we're going to start now. I have 5 minutes to present this one. Yes, sir?
5 minutes? Wow, I hope you don't go that long.
Not to worry.
Is everything working?
Yes, this is working. I just need the citation here, okay? And while you're talking, I'm going to have to move the chair back over there so I can run the timer. Are we ready? Everybody ready? Okay, I'm going to start. Welcome, everyone, and thanks for coming to this symposium in honor of the 2012 Pittsburgh Spectroscopy Award. I'm Joanne Smith from Edinburgh, University of Pennsylvania. I chaired this year's committee and am presiding today. This award honors a scientist who has made significant contributions to the advancement of spectroscopy. The Spectroscopy Society of Pittsburgh sponsors this award, and we're proud to have presented this award to a number of notable scientists, and you see the list of awardees from our first award up to the present on the screen right now. This year's award goes to W.E. Moerner from Stanford University, and we're pleased to have him here today and several of his peers and associates to participate in this symposium. Dr. Moerner has been a leader in single-molecule spectroscopy since its inception, and he continues to make advances with his ongoing research. The Pittsburgh Spectroscopy Awards consists of a scroll and an honorarium, and I'll put the scroll up on the screen in just a moment, as well as the actual copy that you see there. Gregory Meisner, who is the current chairman of the Spectroscopy Society, was unable to travel to Orlando this week, and so Dr. Fume Lin from Dr. Fume Lin, who is the chairman-elect of the Spectroscopy Society, will present the award to Dr. Moerner in just a moment. I would just like to read the citation to you to review his accomplishments. He has been acknowledged as a leader in single-molecule spectroscopy since its inception in the 1980s. Through his early work in the spectroscopy of cryogenic materials, he pioneered single-molecule spectroscopy. Since that time, he has been at the forefront of the technological developments in single-molecule spectroscopy and of its application to elucidating the chemical physics of biological systems and non-biological materials. In recognition of these accomplishments, the Spectroscopy Society of Pittsburgh is pleased to confer this award. So would you guys go over by the scroll, and then if you can make it, and Fume will do that. Okay, Fume, go ahead. Thank you.
Okay, well, let me just say that this is a tremendous honor to be recognized for the Pittsburgh Spectroscopy Award. I want to thank the Pittsburgh Spectroscopy Society for this. It's really a thrilling moment, and I appreciate that very much. And I'm also very happy to just mention that there's going to be 4 spectacular talks later on in this session by former students, postdocs, and colleagues. and so I encourage you to listen for those as well. But single molecules as light sources for super resolution imaging and for probing single biomolecules in solution is my topic for today. And I really just want to turn back the time a little bit and tell you and remind you a little bit about our motivations for working in this area. It's been more than 23 years since the first experiments in my lab at low temperatures with low-tar Cador, 1989. And since then, many, many groups all over the world have started to use single molecule methods, including room temperature and biological systems. And in all of these experiments, we remove ensemble averaging. Okay? We do experiments on individual molecules one by one. This allows us to explore heterogeneity, follow state changes in time, and we can test our theoretical understanding of how systems actually work, but also observe new behaviors that were not observable before when averaging was going on. You can also look on the nanoscale with a single molecule. That's because it's a very small light source, and it can be used for something called super resolution imaging and probing local fields, things that I'll talk about in more detail today. You can even generate quantum mechanical light with single molecules. So it's a huge and very exciting field now after all this time, but since it will be much more fun to talk about what's new, that's what I'll do today. I'm going to talk about several different areas, what's new with single molecules, and in a sense, I'm sort of just doing a warm-up act for the 4 talks that are going to come later on. First, I'll talk about using a single molecule to probe a local electromagnetic field in a special, very tiny metallic nanostructure. And it turns out that you will see fluorescence enhancements. Then I'll talk about how single molecules are being used today to surpass the optical diffraction limit in microscopy. We're using something called single molecule active control. And since this field is right now a very blossoming one, I'll talk a little bit about some examples of how this works and even talk about 3-dimensional methods for imaging with a 2-dimensional camera. And then I'll end up talking about how to look at single biomolecules in solution. Not in cells, but in solution, where they're zooming around normally under the fluence of Brownian motion, but with a device called the Abel trap, you could stop that. And this will involve the discussion of recent work on a chaperonin that's held in the trap. So all of this connects to the 3 speakers that follow, and you'll see as this goes along. So first, single molecules can intimately sense this local electromagnetic interactions. That's because the single molecule is about a nanometer or so in size, and its dipole moment interacts with the local electric field. So to give you a specific example of this, which relates a bit to Callie Willitz's talk later, I want to talk about Anika Kinkabwala's work in my lab, a graduate student, who explored single molecules interacting with something we call a metallic bow tie, a bow tie nanoantenna. These are gold triangles that are only on the order of 70 to 80 nanometers in size with a gap between them on a transparent substrate. And that's one of these structures, the nanoscale structure that has plasmonic enhancements, plasmonic resonances that are being heavily studied in many areas these days. What Anika did was to coat this whole sample with a polymer that contains single molecules in it. Now, the concentration of single molecules is large here. They're coating all over in all kinds of orientations. And so you pump and collect light from this structure and see what happens. We're using a molecule that came from Klaus Mühlen. And here's what you see. Here's an array of bow ties. And all of a sudden, you notice that, gee, where the bow ties are, there's much more fluorescence coming, even though there's molecules everywhere and all over this particular picture here. The ones that are not near the bow tie are dark. The ones that are near the bow tie are bright. How bright are they? Well, it turns out here's a trace from just one bow tie. You see fluorescence emission. This is time. It's been emitting for a very long time, hundreds of seconds, very stable under these conditions. But you see that emission from the bow tie seems to turn off for a second and then down and back up and down and then jumps down to this level, then back up again. This change here, this drop in fluorescence, really can only mean that one molecule just turned off. And since one molecule just turned off, we now know that that molecule was producing 50% of all of the emission coming from that spot. That molecule is incredibly enhanced. And this analysis allowed us to analyze that there was a factor of 1,300 increase in the fluorescence from these single molecules at a maximum when they're perfectly oriented in the right direction, right in the right location. That's coming from both an increase in the local electric field due to the plasmonic enhancements and also due to a reduction in the excited state lifetime because the nanoantenna acts as an antenna. In other words, the electrons from the molecule shape the electrons in the bow tie, and therefore you can couple to the far field much better than you would without the bow tie. So just an example of some of the things that are going on in the single molecule area, connecting them to nanostructures. Now, going on to the second example, because I'll have a lot more detail about this one. Single molecules can provide this route to super resolution imaging far beyond the optical diffraction limit. So this requires a little bit of introduction to make sure it's clear what I mean. As you know, there are structures on many spatial scales of interest to all of us here at this meeting, from single molecules all the way up to cells with bacteria and single proteins in between and so forth. But remembering now, there's this concept called resolution. It's the minimum distance to which two objects can be distinguished in an image. So when we of course, look at techniques like X-ray, NMR, and EM, electron microscopy, those are, of course, extremely high resolution. They work great down in this very, very low scale, but they're not very good for live cell imaging. But they're obviously the best for resolution. But, of course, light is very powerful. Far-field optical fluorescence microscopy works at a distance. You don't have to freeze the sample. You can learn a lot about dynamics as well. It's non-invasive. That's why fluorescence is, of course, a very powerful method. But up until fairly recently, optical light has been limited by the diffraction limit, lambda over 2Na, recognized by Abe in the late 1800s. And so in the visible, this limit is about 200 or 300 nanometers. So that means that objects cannot be resolved if they're closer than that distance. So this new area called super resolution microscopy means optical microscopy where you have extended this boundary down to the 10 nanometer range and below. That's what I mean by super resolution. So it's intending to fill in the gap between the ultra-high resolution methods that don't work on live cells and optics. Another way of thinking about this, let's suppose we're interested in this little bacterium here. It's been labeled. The specific proteins in that bacterium have been labeled with a fluorescent protein. And of course you can't see very much, right? Bacteria are very small, only a couple of microns long, half a micron across. And so you say, well, let's just go down to the equipment show and buy the best possible microscope that we can buy. And so let's do that. Here's that microscope, the best you can buy, and you don't see any more detail because there's this problem called diffraction, as I was trying to describe. So even though the individual labels in this sample are green fluorescent proteins, just a few nanometers in size, when you image that small object, you see a spot on the order of this size of the diffraction limit, lambda over 2Na, a few hundred nanometers in the visible. And so each single molecule looks like a blob and they all overlap. So that's the problem. And this super resolution concept that I'm going to describe allows you to see this instead. So that's the difference between a diffraction limited and a super resolution image. So how do you do this? How can we surpass Abe's diffraction limit, which is a fundamental physical effect? Okay, we're not going to change physics here. So you have to circumvent it in some way, and a way I'll describe. And the method I'm going to talk about here is going to use single molecules plus two big ideas. There are other ways of doing this, of going beyond the diffraction limit, like stimulated emission depletion microscopy or structured illumination microscopy. But today, single molecules plus two big ideas. So, step 0, detect single molecules. That's, of course, the first requirement, pretty obvious. We have to be able to image fluorescence from single molecule labels. Of course, these days, this is very well established. If you have low background and a good emitter, good quantum yield, and a very clean sample, good filtering, and these wonderful new electron multiplying CCD cameras, then we can pump molecules that are fluorescent and collect the emission from those molecules as long as they don't go into dark states too long. And of course, this is the idea that's been pervading single molecule experiments for a very long time. Here's what I think might be the first single molecule image. This is a single fluorophore image from Pat Ambrose in my lab in 1991. There's frequency on this axis, high resolution spectroscopy on this axis, but this other axis is a spatial axis and counts as a one-dimensional image of the object in the spatial domain. Here's a room temperature example from a decade later. And you see that the single molecules, by the way now, in this top-down picture, have sort of a shape. And here's a micron bar. The molecule is not 1 nanometer in size, but it looks like about 200 nanometers wide, a little blob there. So that's this problem called diffraction, as I said. And so the second big idea that you need to get to super resolution is to super localize. recognizing that the image of a point source, or the single molecule on the camera, produces this larger spot called the point spread function. Now, what do you do to make progress? Well, the first thing you do is to say, well, that happens to be a single isolated molecule, so I'm going to spread it over many pixels of the camera. This is essentially important. My camera pixels, you can see them here. What I'm really doing here is to sample the shape of this point spread function. It's not just a little round disk, but it's a little mountain. It has a shape. In one-dimensional cross-section, you can see that it's approximately Gaussian, and this histogram shows you the different samples that I have of the shape of that image of that single molecule. So once we can measure the shape, then you can fit the shape with a model function, a Gaussian function, or more precisely an Airy function. And if you do that fit, then you can find the parameter from that fit that corresponds to the peak of this curve, or the position of the molecule. Now, we can extract the center position of the molecule by fitting that shape, fitting that measurement. That is a parameter that we extract from the data. And so the distribution of measurements or estimates of that center position is far narrower than the width. The width is determined by the diffraction limit, but our distribution of estimates of the center position can be far smaller, far below the diffraction limit. So that's the key idea of super localization. There is mathematics that describes this width here, the variance, okay, of these estimates. And there are terms that depend upon background and depend upon pixelation. But the most important term is this first one, which is the Abbe variance divided by the number of photons detected. All right, that's the key point. Every time I detect a photon on the camera, then that represents an estimate of where the molecule is. So the more photons that I get into this pattern, the better the fit will be, and the better I'll know the parameter for the center. Looking at this primary verse dependence and converting it to standard deviation of this distribution is the Abbe limit divided by the square root of the number of photons. So all of this means that I can super localize the position of a single molecule to much better precision than the Abbe limit. Suppose I had 10 to the 4th photons, then the square root of that is 100. I can localize the center position to, in principle, 100 times better than the Abbe diffraction limit. So that's key concept number one, okay? In addition to being able to see single molecules, we can localize them if they're separated in space. Been used for a very long time in biology and in many other areas of science, fitting the point spread function. But now, what about when you have many emitters? What do you do when you have high concentrations of emitters decorating a structure? Because we want resolution. We want the ability to separate two objects that are close together. And so if I have all of the emitters on at the same time on a structure, and I take a wide field fluorescence image, you would see this blobby sort of thing because those point spread functions all overlap. So the critical big idea number two is that you have to actively control the emitting concentration. So for example, let's suppose that we're going to turn the molecules on and off in some way. There's a lot of ways to do this. But let's suppose, for purposes of argument, let's suppose that these molecules are not on all the time, but let's make them photoactivatable. Let's make them dark until I use a little activating light to turn on a few of them, okay? So here they are all off, so I don't see the structure at all. And then I use a tiny low-power activating light, and you can make that arbitrarily low so that only a few single molecules will turn on. And so, for example, here's 5, and when you have them when you take this image, then you can fit each one of those single molecules and find the center position. And then you photo bleach them, use them up, and do it again. Turn on a few more. And so then you will get more positions, more samples of the underlying structure. With the final result being, after doing this a bunch of times, you get what's called this super resolution image, which is like a pointillist reconstruction of the original structure. Okay? But it really is single molecule sampling along the structure by the scheme that I've been talking about, controlling whether the emitters are on and off. Now, this idea appeared on the scene in about 2006 in 3 groups and with 3 different acronyms. PALM, Photoactivated Localization Microscopy, Eric Betzig, but also Xiaowei Zhang's lab, something called STORM, Sam Hess, FPALM. The key idea and then there's many more after that. Something called PAINT, D-STORM, a YFP reactivation we worked on, but without an acronym, GSDIM, another acronym, BLINK, another acronym. The point I want to make is that there's a general concept overlying all of these schemes, and it is that you must use some sort of optical control or other kinds of control to keep the emitting concentration at any time low so that you can do the single molecule localization. And because of the unity of all these ideas, we like to call this, by this mechanism-independent phrase, single molecule active control microscopy, or SMACM. So that's the basic idea that is being used to surpass the diffraction limit using single molecules. Now I want to give you some examples. So first we need to have a fluorophore that's controllable. The fluorophore that I'm going to use is the one that Rob Dickson studied in my lab back in 1997. These are actually the first experiments to image a fluorescent protein, and it was enhanced YFP, yellow fluorescent protein. We got the molecule from Roger Chen's lab. And what Rob saw from the very beginning was some really interesting surprises with this fluorescent protein. First of all, he saw blinking. He saw that the molecules will emit, frame after frame, and then turn off, but then come back on, and so forth, which we interpreted as cycling between the ground state and the excited state with emitted fluorescence, but every once in a while entry into a dark state from which you can return thermally and therefore start emitting again. So blinking is present in YFP, but sometimes you would also, after extended irradiation, see them turn off completely. And what Rob also found is that another color of light, a shorter wavelength, would turn the molecules back on. Here's a used up molecule, a restored molecule, used up, restored, etc. And this whole scheme has been also shown in cells by Julie Biteen. So the point here is that you can use this for this kind of imaging I've been talking about. All you need to do, for example, if you're thinking of blinking, is to turn the power up so that almost all the molecules are in the dark state. And then a few turn back on, but only a few at any given time. And you can localize those and therefore use that to build up samples of an underlying structure. So you could use activation, you can use blinking, etc. So moving to an even more specific example, let me just show you what these data look like now. For EYFP, EYFP has been fused to a bacterial protein in all of these bacterial cells here. You can see them in this white light image, and this is a single frame of the movie. But if I run the movie from the camera, you see this fantastic blinking on and off of the yellow fluorescent proteins, which are fused to a specific structure, a specific target protein inside these cells. And so this is the kind of data that people analyze in this field these days. Each time you see a single molecule in a given frame, you fit it, find its center position, and do that again and again and again to build up data on the underlying structure. Typically, these experiments give resolutions that are down to the 30 or 40 nanometer range, even with fluorescent proteins. All right, so specific examples, okay, of this idea. Here's a wonderful slide produced by Steve Lee from my lab that reviews several different experiments like this in the recent history. These are all epifluorescence images. And in each case, there's a bacterium which has been, in which a particular protein, in this case MreB, has been fused to YFP. Here PAR-A has been fused to YFP. Here HU has been fused to YFP. And once again, diffraction-limited imaging doesn't tell you very much. They all look pretty similar. But when you use SMACM, when you use this single molecule palm storm method, you see this fantastic array of different protein superstructures inside these cells. So we've now gone from this regime, diffraction limited, to super resolution. MreB forms a helical-like structure. PAR-A is a filament that runs right down the center of the cell. And then HU is a protein that binds nonspecifically to many regions of the DNA. So it represents a sampling of DNA locations. So this is kind of where the DNA is inside the cell. And each of these experiments have, as I said, a resolution of about 40 nanometers or so. The neat thing about PAR-A is that by doing these experiments, we observed that it was a filament for the first time. Previous to this, only these kind of measurements were available. And PAR-A turns out to be the track, if you like, that the newly synthesized chromosome of the bacterium moves from one pole to the opposite pole before cell division in the center. So it's the bacterial division spindle. We have many spindles in our eukaryotic cells. This bacterium has one, right down the center, about 40 nanometers in width. So that's some examples of what you can do with this whole scheme. Now, you don't have to just use blinking of fluorescent proteins. You can also use other molecules, fluorophores, that are controllable in some way. And so in collaboration with the Twig Lab, we've been working on photoactivatable fluorophores, small molecules, small organic molecules. They're great because they give more photons than fluorescent proteins. I don't have time to tell the whole story, but this is the fluorogenic form of the molecule, which with light you can convert to the fluorescent form of the molecule. And so, again, as I showed before, if you have this decorating a structure using, for example, a halo tag targeting moiety, then you can use weak activation to turn on singles and then image all those singles to get super resolution. This is an example from the work of Xiao Li in my lab. This is the diffraction limited image of microtubules in BSC1 cells. This is the super resolution image of microtubules in BSC1 cells generated by using this particular small molecule fluorophore. So that just shows you that there's many different now ways of doing this. Many different blinking emitters can be used to get super resolution by these schemes. Great. So now what about 3 dimensions? What about 3D? Everything I've showed you so far is 2-dimensional. We'd like to extend to 3 dimensions. And we're doing this in a special way, but let me just set up the problem for you. Look at this set of images here on the left side. This shows you what a conventional microscope does in terms of its point spread function, or you can think of a confocal microscope if you like. And what I'm doing is highlighting that at many different z-planes, if I have an emitter at different z-planes relative to focus, of course it's nice and bright when it's in focus, but it gets dim when you're out of focus, etc. But the real problem here is that this particular response has very little information about the z position of the emitter. Okay? This function has 0 derivative at the focal plane and only smoothly goes off on both sides. It's also symmetric above and below the focal plane. It doesn't tell you very much about z position. There's been a variety of schemes that have been used to get around this. My lab has been pursuing something called the double-helix point spread function. What we do effectively is to change the microscope so that it has a different response. Instead of a single point emitter having only 1 spot, the single point emitter has 2 spots, and the 2 spots revolve around one another as you move to different z focal planes of the emitter. So this is an image of a single molecule. Okay, actually, in this case it happens to be a fluorescent bead. But as you move the sample up and down along z, you see that the 2 spots rotate around each other. So if you have this sort of behavior, all you have to do is to calibrate with a bead while moving the stage in a well-defined fashion. And in other words, you can find out what angle corresponds to which z. Then you also, in each frame, you fit both lobes, and those 2 lobes will give you 2 positions. You take the midpoint between the 2 of them for x and y, and you take the angle for getting z. So this is a way of encoding z into the image itself. It works by some wonderful optical magic. This is the result of some beautiful work by Raphael Pittcon at the University of Colorado, which we've adapted to the single molecule problem. And I'm showing you here a rough sketch of the optical setup, just to show you how much you have to change to get the double helix response. Over here is a conventional inverted microscope. There's the objective, here's the sample, and here's a tube lens that will give a real space image out here. And I have a sample here with 2 single molecules in it. Okay, so there's molecule number 1, molecule number 2. That's the standard point spread function. So we don't change anything in that part of the microscope. In order to achieve the double helix response, what we do is Fourier transform this image using a 4F optical system. And at the Fourier plane, there is a spatial light modulator which changes the phase of the light, or multiplies the Fourier transform of the image by a phase pattern that corresponds to the double helix response. Then you Fourier transform again back to real space. And so single spots become double spots. In effect, what we're doing is convolving the standard microscope's response with the double helix response. And this happens to be the phase pattern that's loaded on to this array. So it's a beautiful piece of optics. There's some sort of magic, if you like, such that even though I don't know what the z position of these 2 molecules is from this normal image, here I can tell their z position because the pairs of spots are rotated at slightly different angles. So this scheme we've been exploring heavily in my lab and using it to do some interesting science. One example is to track single objects. And Mike Thompson, working with Pat Brown's lab, tracked a single mRNA in a living yeast cell in 3 dimensions using the double helix point spread function. Here again, it's showing you that for different z's, the 2 spots will rotate around one another. And so this yeast cell has a particular single mRNA labeled with a bunch of fluorophores. And let me just show you what it looks like if you now look at the movie in the double helix microscope. You see there's two spots there. That's because the double helix is on, and you see them rotating around one another. That's because that single mRNA moved in X, Y, and Z during this experiment. So we're doing this with 15 milliseconds time resolution and 25 nanometer XY precision and 50 nanometer Z precision. Bottom line, two-dimensional images become now, two-dimensional tracks become these fascinating three-dimensional tracks over a large spatial range. This is 1.5 microns along the Z direction here. And there's a great deal of detail hidden in a trajectory like that, corresponding to sometimes directed motion, sometimes diffusion, binding to objects, and so forth. So the double helix provides an interesting way to get this three-dimensional information from tracking. What about imaging? Well, you can also image extended structures. Here I'm just pulling one example from our bacterial experiments by Matt Liu and Steve Lee. These are bacteria, Caulobacter crescentus, from Lucy Shapiro's lab, where we're essentially doing a two-color experiment. Inside each cell, you can see something in orange and red. That turns out to be CRES EYFP, or the crescentin protein, labeled with the yellow fluorescent protein. And it's imaged by the blinking that I showed you earlier, but in 3 dimensions now. And then the cell surface is being lit up by Nile red, a paint dye from the Huckstrasser idea that came some years ago. It's lighting up the cell surface. So this is a 3-dimensional image of the cell surface and a single protein inside the cell, forming an interesting sort of quasi-helical superstructure. And it just gives you an example of what you can do with the double helix. We also can use the double helix in mammalian cells, but I don't have time to show you that example right now. So that's the double helix and super resolution provided by single molecule imaging. The third area I'd like to review to give you a flavor of some other interesting things that are going on with single molecules relates to trapping single molecules in free solution for detailed spectroscopic analysis. And so this requires a little bit of introduction to something that I'm going to call the Abel trap. You'll see why in a minute. I'll introduce the Abel trap, but I'm just showing you here that we've now applied it to a large number of interesting biophysical systems. And Randy Goldsmith will be talking about fluorescent antenna proteins and a redox enzyme in the Abel trap. I'll talk about this particular one, which is a multi-subunit enzyme. But we've also looked at single G-protein coupled receptors in the trap, and student Tran Wang is developing new and advanced trap designs. So what is this trap device, and really why do we need to have this? Why do you think that we must have it? The goal here is to avoid surface attachment, okay? We don't want to have large beads, so we're not talking about laser tweezers here. I'm not talking about optical forces or anything like that, where there's a really large bead that's trapped with a high-power focused beam. And we don't want to encapsulate the single molecules because we'd like for them to be as native as possible. At the same time, we want to observe the same single molecule as long as possible to extract some detailed dynamics. Of course, the real problem is that these biomolecules are small, on the order of, let's say 10 nanometers. And so they're buffeted by Brownian motion in solution, of course. Well, why not wait then for diffusion, as in fluorescence correlation spectroscopy? Here in FCS, or fluorescence correlation spectroscopy, you focus a laser beam into a liquid, and you wait for molecules to diffuse through that focus. And then you get a burst of fluorescence and so forth. An exciting area. Has a lot of people have worked in this area to get interesting information on short time scales. But you get only a brief burst from each molecule on the order of about 1 millisecond's worth of observation time. In addition, the intensity is non-uniform. This Gaussian focus spot has a Gaussian shape. So if the single molecule diffuses through the center, you get a bright burst. If it diffuses through the edge, you get dim bursts. And so you don't have much amplitude information from FCS, but mostly time information. And also, Hao Yang has worked in this area, but tracking to find a single molecule. Here, we want to trap the single molecule. So we need a better idea than FCS. So the idea is something that we call the anti-Brownian electrokinetic trap, or Abel trap. Something that Adam Cohen built when he was a graduate student in my lab, then a postdoc, and he's now a professor at Harvard. Here's basically how it works. we first of all detect the position of a single fluorescent particle. Now this is just imaging, so I've already talked about that. We can image single molecules, but once you've imaged it, then you know something about its XY position. Then what you do, if it's not where you want it to be, then an external feedback loop applies forces to move the object back to the target. So what this really means is once you put on this force, then the molecule starts diffusing again and moving toward the target. But next time you look at it, it's somewhere else. It's not quite at the target. So then you need to produce a new force in that direction, so it starts diffusing in that direction. Next time you look at it, it's here, and so forth. So if you close this in a feedback loop, okay, high-speed feedback loop, then the result will be just that the single molecule will jiggle around the target location. And in that sense, because you're illuminating a big region, you can observe it for a long time. So every time a Brownian kick moves it away, we kick it back, and so forth. So this is done inside a microfluidic sample cell. Here's a sketch of the cells that we've been using. And all of this action I'm talking about is right here in this central region, about 10 microns by 10 microns in X and Y, but about 700 nanometers thick in the Z direction. We're confining the motion in Z, so we don't worry about Z position or Z tracking here. And all of the forces I'm going to talk about are being applied in the XY plane by putting voltages on the solution at large distances. So the forces are electrokinetic forces, and primarily in these experiments, they're electroosmosis forces, all right? They work for objects that have any charge, and including uncharged. And so that's the basic idea, though, and if you make this all work, you can trap single molecules, certainly single labeled biomolecules, and all the way down to a single fluorophore in recent work in the Cohen lab. But the key idea here is that this is very different from laser tweezers. Laser tweezers requires a big bead, as I say, and laser tweezers scales with the radius of that big bead cubed. This experiment, if I make my object a factor of 10 smaller, all I have to do is to run the trap a factor of 10 faster, and I'll still be able to trap the object. So it scales linearly with the object's diameter or radius. So you can trap very, very small objects, scales more favorably than laser tweezers. Okay, so what's an example of how the Able trap works? I'll talk about the revolving beam implementation. There's a number of different ones, but here's a top-down view of the microfluidic cell, and here's the optical apparatus, which is pretty much like a confocal microscope, except for some changes. First of all, in the revolving beam incarnation, an acousto-optic deflector is used to rotate the pumping beam in a circle like that. So the pumping beam revolves around the center of the trap with a small radius. And that's the way we're going to sense position, because when a single molecule is present right there off the center, it's going to emit a photon, okay, when the laser beam gets to it. And because we know how the laser beam is rotating, then we're able to know what direction that single molecule has gone off the center, so you can put a force on to drive it back. That information is extracted from the phase of the modulation or from the time at which the photon was emitted. And so the net result then, something like a lock-in amplifier in electronics, then apply the force that's required to push the object back to the center of the trap. Okay? So the rotating beam is nothing more than a sensor of position that lets you make the trap work. And here's a single GRO-EL chaperonin protein that's got a few fluorophores on it that's trapped in this trap in free solution. So I want to describe one quick experiment of the Able trap worked by Yan Zhang on a chaperonin, but the chaperonin here is a little more interesting to us. This is TRIC, the chaperonin enzyme TRIC, which is essential in all of our cells for the folding of actin and tubulin. It is a double-barreled enzyme. There's essentially two rings stacked on top of one another. There's 8 subunits here and 8 subunits there, a total of 16. And each of the subunits can bind and hydrolyze ATP. So the question of interest here is, what can we learn about ATP binding and hydrolysis by using the Able trap on this system? Well, to make that work, Yan had to make a fluorescent form of the TRIC. And in particular, we decided to make the ATP itself be fluorescent because we want to count the ATP. So this molecule is a fluorescent form of ATP where a Psi3 dye has been added to it. The chaperonin is incubated with that ATP, and then after some time period, it'll hydrolyze and inorganic phosphate will leave, and you'll still have ADP that's fluorescently labeled at the end. And so once you then purify away any unbound ATP, you have chaperonins that have a certain number of ADPs that are fluorescently labeled attached to it. So if you take those objects and put them in the Abel trap and see what you see single chaperonins coming in, and then another one coming in, another one coming in, another one coming in, et cetera, et cetera. And let's blow up each one of these. Here's one, we blow it up. And you see very nicely that for that single chaperonin, the photobleaching goes down in steps. That is, the different ATPs that are on the system, that are on the chaperonin, are photobleaching, but because we're able to sort of watch these steps, we can count how many ADPs were bound to that single chaperonin. That's the key variable we're picking out of these experiments, the number of ADP. So this allows us to make a new kind of measurement on single molecules, the ADP number distribution. And we plot the number of ADP on distribution-like curves that are common in the single molecule community. This particular form of displaying it is a special form that is just designed to give you information about the errors in our knowledge of how many ATPs were on each chaperonin. There's a histogram and then a probability distribution. The thing you want to look at is the red curve, which is the overall probability distribution of ADP numbers for single chaperonins. Okay, so great. What can you learn from this? Well, let's watch it as a function of time. Okay, we're going to purify away unbound ATP, and then rush to get the molecule in the trap, the molecules in the trap. And here's the distribution. Okay, it's the same as the red curve I just showed you, just plotting it in blue now. It took 10 minutes to get the sample into the trap, and you see that all of the chaperonins that you observe have essentially still 8 ADP on them. Okay, that's interesting. Now let's go a little bit later. 15 minutes to 20 minutes later, still 8. 20, 25 minutes, still 8. So amazingly, what's going on here is that we don't see 6, 4, 2, and so forth. But what we see is only chaperonins that have 8, because when it decides I'm going to get rid of my ADP, I'll get rid of them all at once in a very highly cooperative fashion. So this proves that there's extremely high cooperativity, because you don't see any cases that have smaller numbers. Very interesting result, very synchronized ADP release. The integration of these curves gives you a single exponential fall off that gives you an ADP releasing rate that matches very nicely to a bulk ADP releasing rates. So bottom line, this is just a way to use the Abel trap to learn something new on multi-subunit enzymes, to count the number of nucleotides that are present under different conditions. There's other work, but I don't have time to describe it. So to summarize, okay, this wonderful event today, I've talked a little bit about some of the new things that go on in single molecules. Interactions between single molecules and nanoscale metallic structures, this whole interesting area of super resolution to go beyond the diffraction limit in 2D and in 3D. And this Abel trap is a nice way to look at single molecules in solution for very detailed analysis. All this work would not have been possible without my wonderful group of students and postdocs and collaborators. Over the years, I've only got the current group here, but you're going to hear talks from several of the people in the list of alums here, and also from Hao Yang, a friend and collaborator. And with all of this, again, I want to thank you for this award and urge you to stay and listen to the other 4 wonderful talks coming up.